Our next student is Jacob. He's the first of our two programming interns. And something special about Jacob is that he's the only rising junior of the class. And if you have been following the program for a while, you know that it's already very hard to get in. But if you are a rising junior, it's very, 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 very hard. So in the last 5 years, we only had 3 rising juniors make it. And Jacob did a fantastic job. He's an incredible programmer. And just an example to make you feel a little bit bad about what you were doing in high school. He already presented some of the results that he got last summer doing research at the annual American Society of Physics. Jacob?
Good morning. As Ilari just said, I'm Jacob Rybinki from Suncoast High School. And this summer, I worked at the Bidet Lab as a summer programming intern. And my main project was developing a program that automated the process of our data collection and analysis. So before I really jump into my presentation, I want to go over a quick outline. So I'll start by really posing the question of how do we move, how does movement work, and going over what we're actually studying at the Bidet Lab. Next, I'll jump further into one of the current experiments that we're running and our experimental setup. Then I'll get more further into the programming and some of the machine learning tools that we're using. And finally, I'll really go granular into my pipeline and what processes I actually automated. So I want to begin by posing the question of how do we actually move? How does movement work? So although from a very broad perspective, this might seem like a very simple question because, you know, most of us learn how to move when we're babies, and to most of it, this feels like a very natural process. But from a deeper perspective and from a neuroscience perspective, this actually becomes a very interesting question. So the main thing that we're trying to understand at the Bidet Lab is how does neurons firing in the brain lead to this kind of coordinated movement, especially like how we move around, how we walk around. So I want you to note that this is a periodic motion. So when we walk around, when we move around, we're repeating the same action, a similar action over and over again. It also exhibits a variety of patterns. So depending on our speed, on our direction of movement, or even on the terrain around us, we can change how we're walking, how we run, how we move around. And finally, it's not a fully conscious process. What I mean by that is that although it seems like we're in control, our brain is doing a lot of the heavy lifting. Because even when we take a single step, we're activating a lot of muscles in our legs and our entire body, and our brain is keeping us upright, keeping us moving, and keeping us balanced. So it's not as simple as it might seem to us at first. So as you know, at the Max Planck Institute, we're not studying humans. We're not like cutting open people or experimenting on humans. So what could be the next best thing to humans other than fruit flies? Now, although from this initial kind of take that you have, the fruit fly might seem maybe not nuanced enough to be able to answer a question like I just posed. This animal is actually very interesting, and it provides us with a lot of tools, and it gives us a new ability, or a very good ability, to understand movement and how the locomotion works. Now, one of the main reasons for this is actually because it's more simplified. So whereas the human brain has 86 billion neurons, the fruit fly brain has orders and orders of magnitude less at just 100,000 in its brain. And this reduction in neuron count vastly increases the easiness and the ability that we have to actually study its brain and understand what the neurons are doing. And second of all, even though it might seem from the outside to be like a very simple animal, it actually still exhibits a lot of these complex functions, especially in terms of locomotion and movement, that we need in order to be able to understand and study the question of movement. So they exhibit many complex behaviors because unlike us, where we only have 2 legs, they actually have 6 legs. And even just this one thing makes it so that there's many, many more combinations of how they can use each of those legs in different movement patterns, which are officially called gaits. So if you look at this picture right here, we can see there's a sort of tripod gait, which as the name suggests is when 3 legs are on the ground. There is a tetrapod gait, where the fly is using 4 legs on the ground at the same time. And then there's this whole group that we just call non-canonical, which is just the lump of all the other possible movement combinations of the fly. And it's not picking any of these and sticking with it. Just like we can switch how we move, as the fly moves around, as the terrain changes, as its speed changes, it can continuously switch between all of these different movement patterns. So now that we broadly understand what we're using to study this movement question, how are we actually going to do this? What kind of experiment do we need? So chiefly what we need is, first, an easy way to stimulate the neurons. This means that we want to be able to very easily turn on and off these neurons and be able to identify neurons that we want to study and be able to activate or inhibit them and do whatever we want with them. And the second thing that we need is to be able to target specific neurons. So again, owing to the more simple structure of the fly nervous system, we have the tools available that we can target very small groups of neurons or even just individual neurons in its brain. And finally, we want a technique that's non-invasive. So what that means is, as you can see, we don't want to be cutting into the fly, and we don't even want to be really physically interacting with it very much, because the more we're interacting with this fly, the more that's probably going to affect its movement, and then it might muddy up our data, or it might make it walk unnaturally or move around unnaturally. So our main answer, our main method of experimentation to cover all three of these points is something called optogenetics. So as the name suggests, opto, we can activate individual neurons or activate the neurons we're interested in by just shining a beam of light at the fly. So owing again to the simplicity and the tools we have available with the fly, we can actually genetically modify the neurons that we're interested in to be sensitive to a specific wavelength of light. And then when we shine that light, it can pass through the kind of exoskeleton of the fly, through its head, and then hit those neurons. And the neurons that we've modified will actually start to activate due to the response from that light. And this is also this can be a very specific activation, as I mentioned. So to give you an example, we can make the fly do these very, very specific actions. For example, we can make them run forward, we can make them move backwards, or we can make them turn around. So these are all very specific actions that we couldn't actually do in a more complex animal. But in the fly, individual neurons or very small groups of neurons can actually control these kinds of movement actions. So this is the general background on the experiment. Our actual setup looks something like this. So if you look just in the middle, if you focus on the middle here, there's this little tiny ball that the fly actually stands on. And you can kind of imagine this to be like a treadmill. So again, we want to be non-invasive with the fly. We don't want to affect its movement too much. So what we want to do is have a very frictionless kind of plane that it can walk on. And on this ball, it can walk in any direction endlessly. And the ball is suspended on a stream of air, which means that there's minimal friction. And if you look around it, surrounding the fly, there are 8 high-speed, slow motion, high frame rate, very high-quality cameras. What those do is that they capture every angle of the movement of the fly at very high resolution and a very high frame rate, so we don't miss a single data point, we don't miss a single motion of the fly. And this, again, allows us to recover the movement data of the fly just from these videos. So we don't have to attach any trackers to the fly, we don't have to be touching it, and so on. And over here on the right, you can see there's this little rod coming in. One half of that is actually the cable that's shining light on the fly. It just directs the light onto the fly, which is what's actually activating those neurons. And then the other part of that is our only physical point of contact with the fly, which is a little cable or a little rod that we attach to the fly so that, of course, it doesn't just fly straight off of our experimental setup. Now, we have all this data from our experiment. We have all of these videos. How are we actually going in to process that? So one of the main kind of programmatic approaches that we're using is called machine learning. And the first tool that we're using, which you may have heard about in Lydia's presentation, is actually called Deep Lab Cut. Now, in her presentation, she mentioned its use on mice, and this is kind of a similar example where we can see it tracking mice. And obviously in our lab, as you can see, this is an example video of what the experiment can collect. And Deep Lab Cut automatically will go in, and it'll track the limbs of the fly that we're interested in. So what this is really letting us do is save hours and hours of manual labor, because previously, before such tools existed, somebody would have to go in and manually go frame by frame, marking up all these points. But now we can, with very high confidence, just be able to give Deep Lab Cut all of these videos, and it'll go in frame by frame and automatically output to us these annotated points. And the way we're telling it how to do this is we do something called training. So just like how you might train a human by showing them examples, we show this computer program hundreds of examples of human annotated examples of frames of, let's say, the fly walking. And after a few hundred times, the program will learn exactly what we're looking for. And when we feed it in this new data, it'll now understand what we want to see out, and it'll annotate the fly like this. The second program we're using extends on top of Deep Lab Cut, and this is what lets us get a model of the fly. So it's a program called Anipose. It takes in all of those camera angles that you saw that have been processed by Deep Lab Cut, and it turns this 2D representation into 3 dimensions. So just like you or I, we see this 2D image from our eyes, and we can recreate this kind of 3D representation of the world. Anipose basically looks at all 7 or 8 of these angles, and it'll give us back this 3-dimensional model, which we can, if we want, visualize, like you see here in this video, of a very similar video, except now we have this 3D reconstruction of the fly. So once we run these programs, how are we processing through all this data? What is the current pipeline? So we get all these 8 videos at very high frame rate, very high resolution, and they're really massive video files. So these are like dozens of gigabytes, and we can't really work with that, so we have to compress it down. So we compress it down without losing any critical information so that we can really manage them better and be able to store more videos. Next, we pass it in, as I said, to Deep Lab Cut, which for every frame of every one of those 7 or 8 videos will give us back these annotated points for all of these videos. And then we pass it into several scripts and several sub kind of programs that we've written that'll pre-process this data. So what that means is it'll convert them to the correct format, remove any artifacts that the model might have generated, and create all of the necessary file structure for Anipose, which also involves getting in all the configuration files, creating any folders or file structures necessary, and really choosing all of this manually. And then finally, we pass it into the last step, which is Anipose, which, as I showed, will give us back this 3D model. So what are some issues that we're currently having? As I mentioned, all of this right now is basically manual. So when we're switching between these programs and when we're moving in all of this data, all of those steps have to be run manually. So all of these pre-processing scripts, dragging in all of these configuration files, creating the file structure that has to be very specifically made for Anipose takes a lot of time. And one of the most time-consuming steps actually is that all of these like settings and configurations change with respect to when the experiment was run or how we calibrated it. So somebody has to like think through, when did I run this experiment? When was this video made? And pick the right file. And if we make a mistake, then we have to start all over again. So all of these steps, all these manual steps, not only take a long time, but also introduce a big possibility of human error or bias. And this all adds together so that our current workflow just can't keep up with the pace that we're generating videos and experimental data. So right now, we've only processed about 1/3 of the data that we've generated. So what I basically made is a program that does all of the similar steps. However, it runs through them all automatically. So if we see here, the only input that it's taking in is the file path, so basically where you're storing all of these videos, all this experimental data. Then it'll automatically run Deep Lab Cut, automatically grabbing the correct model, the correct files that it needs for the configuration and all the settings. It'll automatically configure all of this. Then it'll automatically run those pre-processing scripts, which generates the correct file structure, it cleans any artifacts, and so on. And then finally, it'll run Anipose on it and give us back just what we want. So now whoever is processing this data can just put in where the data is, click basically one button, and it'll output to them exactly what they want, which is that 3D data. So as I mentioned, some of the implications is that this vastly speeds up our experimental process. So once you finish an experiment, you can start automatically with one click running this, this program, which will automatically run through that pipeline. It'll let us finish experiments much, much faster. And to really put a number on that, it'll save us around 1.5 hours of work per day of experimentation. So if you think about that over weeks and weeks of running this experiment and collecting more data, by the end of it, we'll save dozens and dozens of hours. And finally, it'll greatly reduce kind of the error and possibility for bias because we're going from dozens and dozens of steps that each one might have a possibility of error to just basically one click and then you'll get the correct output. So what this will really let us do in the future is facilitate more data collection and allow us to start expanding our experiment. So really some next steps for our experiment is to make it much more granular. So as I mentioned, our current setup is a very binary approach. We can either turn this light on or off, which will turn on and off the corresponding neurons. But some of the future ideas that we have for a future experiment is to actually measure the individual activation level of each neuron. So one way we can do this is to use a similar approach of genetically modifying the neurons we're interested in, but now they'll actually emit light based on how active they are. And if we combine that with our current setup, which is measuring the kinematics and the movement of the fly, we can really combine those two data sets and really start to understand how individual neurons firing, how the individual activity of a neuron will lead to very, very specific movements in the fly. And this will allow us to further study and really start to understand how these neurons are working. So with that, I want to thank our lab leader, Dr. Bedea. I want to thank my mentor, Dr. Spiller, who helped us out with this project, and guided us through. I also want to thank Divya, who helped me a lot with making this presentation and understanding the science behind this. And I want to thank Ilaria and Erica, who helped facilitate this internship and are in charge of all of the outreach at Max Planck. Finally, I want to thank my fellow interns and all of the MPFI supporters. And I'm now open to your questions.
Questions for Jacob?
So what did you use to train the machine learning model based off of the human-made annotations? Was that performed in Deep Lab Cut, or was that done in a different program, or how does that work?
Yeah, so the question was essentially, what is the process of training? How are we actually giving Deep Lab Cut that training data, and how is it learning this? So yeah, Deep Lab Cut, because it was developed, for the use of scientists, they actually made it very simple to kind of just give it the data, the annotated data sets that you have, and then it'll train the model, and then we can later use those models. So the pipeline will actually select the correct model that we've pre-trained for Deep Lab Cut.
Any other question? Thank you so much. Great job.